Thank you, Rachel. All right, so today's talk will be epilepsy genetics, and I'm going to go through some basics of genetics quite a bit because I think it really helps in understanding what we're looking for and what we're looking at. So apologies in advance if this starts to feel a little bit like high school biology. I have no disclosures. Nobody pays me for anything, so I'm not biased in any way. I'm going to start by talking just real essentials, like an overview of epilepsy in terms of how it relates to genetics, and then the basics of genetics. And then I'll talk about what kind of genetic testing is, like what tests we do, what kind of results you can get from those tests. And then I'll end with a conversation about the risks and the benefits of testing. So starting with the basics, causes of epilepsy. So when someone says, why do I have seizures? Why do I have epilepsy? I think about some broad causes and reasons that people can develop epilepsy. Worldwide, probably the most common cause for epilepsy is actually infection. And then after that, people can have a structural brain abnormality that they were born with that can cause seizures. Sometimes the seizures start early in life, sometimes later in life, but usually it'll show up on some imaging why that person has epilepsy. And then another big group of causes of epilepsy are brain injuries, which is kind of a broad topic. Like that could include strokes, bleeding, birth trauma, or other acquired traumas. Tumors are a common cause of epilepsy, particularly later in life. People can have blood vessel abnormalities, funny blood vessel formations, either they were born with or they acquire later that can cause seizures and epilepsy. Rarely, people can have autoimmune conditions where the body actually attacks itself in a way that irritates the brain that can cause recurrent seizures and epilepsy. And so where does genetics fit in with all this, right? I just listed a bunch of causes that I didn't mention genetics. And that's because genetics really underlies a lot of these things. So congenital structural abnormalities can be genetically caused. Tumors can actually be caused by genetic risks for those tumors. And then there's big bucket categories that I just thrown at the end. Other genetic causes. There are some epilepsies we know are genetic. And then there's the big unknown. A lot of people have epilepsy. We don't know why. And genetics probably plays a role in many of those people, but we don't have a name for it. We don't have a label for it, but we suspect it might be there. In terms of types of epilepsy, for this talk, I think the best way to kind of lump epilepsy together is to think about it in these categories. Symptomatic epilepsy would be epilepsy secondary to a known brain abnormality. Something's wrong in the brain. It's not working the way it should, and that problem leads to recurrent seizures. This problem could be acquired in life, secondary to head trauma, or it could be genetic and present there lifelong. Sometimes symptomatic epilepsy is in the form of a syndrome, and what a syndrome means it's a collection of symptoms and a collection of maybe types of seizures or patterns of seizures where you can see it happen in multiple people the same way. So it gives you an idea of what to expect in the future and what other symptoms to look for. And then there's a category of epilepsy called the developmental and epileptic encephalopathies. What these refer to is a group of severe epilepsies characterized both by seizures, which are often really hard to control, as well as encephalopathy, which is a term used to describe the really significant developmental delay or even a developmental regression. On the other side of things, there are the idiopathic generalized epilepsies. These are people with otherwise healthy brains that just happen to have seizures. We actually assume that these are probably all genetic epilepsies, but they're largely unidentified, meaning I can't tell you what gene is causing it. And I think that's probably because a lot of these, or most of these, are polygenetic. And I'll get into a little bit of this later, but you need multiple different genes to give yourself the risks of that epilepsy. Then we also think of idiopathic focal epilepsy. So like the idiopathic generalized, these people have otherwise healthy brains, but instead of seizures coming from everywhere all at once, focal seizures start from a small area. And we know of some of these that are genetic, or often these may relate to underlying structural findings too. So now to delve into the basics of genetics. Here I have a picture of a human cell. Okay? Flashbacks to biology. And at the center of the cell is the nucleus. The nucleus of the cell is where genetic material is stored. And the nucleus is responsible for regulating those genes and having them replicate, processing the genes, and it also coordinates the cell architecture. So I kind of think of the nucleus as being the brain of the cell. But the cells are what make up our body tissues and our whole body, and we're made of cells. So when we're talking about DNA, we're usually talking about the DNA that's in this nucleus structure in our cells. All right, so you've probably heard the term chromosomes, and because of the company 23andMe, we all can remember now that the human genome, there are 23 pairs of chromosomes, giving us a total of 46 chromosomes that will be present in every single cell in our body. 22 of these chromosomes are called autosomes, okay? So that means they're the same as in males and females, and they're numbered here by size. You can see number 1's big and number 22 is the smallest. The 23rd pair of chromosomes are what's called the sex chromosomes, so they're different in males and females. The females will have an X and an X, and a male will have an X and a Y. And this pretty picture of all the chromosomes lined up, this is called a karyotype, and it's one of the forms of genetic testing that I'll talk to you about in a few minutes. So those chromosomes that I showed you are made from DNA. Now you might be familiar with this double helix structure that you see on the left. What that is it's made of base pairs. You'll see adenine, thymine, guanine, cytosine. They're linked up in the center of that structure, and it goes up like a spiral staircase. And this is any organism's genetic material is stored in this structure. Taking a closer look of what that double helix is made of, this is just a picture of the nucleotide bases, just to show you on an anatomic level, they're made from elements. So we're talking about really, really, really tiny things. Every chromosome has millions of base pairs in it. So millions. All of our DNA, all of those millions of pairs, that makes up the genome. Okay, so the genome is all of it, every single one of those pairs in that form divided into chromosomes. What an exome is the coding for genes. And this is, genes make proteins, so this is kind of where we think the bulk of the important information lies. That is actually only 1.5% of the entire genome. So a very small amount of the genome is actually coding for those proteins. To try to make this understandable, if you think about the genome as being like watching a football game from the very, very, very beginning all the way to the very end, the exome would kind of be like reading the game highlights in the newspaper the next day or on the internet the next day. It's really just the take-home features. Not every detail, but it's the biggest, most important details. So again, the exome codes for genes, and each exome is then made up of exons and introns. And those exons taken out, put together, will actually build the protein. That's the coding will build the protein. And the introns are extra coding in between. So of all of our DNA, most of it isn't being used for the actual coding of the gene, but all the extra coding probably influences things like how often a protein is made or when that protein becomes active and is made more or less. So what's a mutation? So a mutation is just a change in that DNA. What scientists have done is they've analyzed DNA from probably millions of people at this point in time, and they find out, these are the common ones that we see in everyone. And then a mutation would be a difference that you don't see commonly. So it's thought to be different from what most people have. And mutations happen, they're random, and they happen all the time. You can't stop them from happening. You can make them more likely to happen. A good example of what can make a mutation more likely is radiation exposure, whether it's from the sun or a flight or therapeutic radiation for disease. It causes mutations to be more likely. A mutation could be good. So we think that's human evolution over the years. A random mutation will happen, it'll help the species, and so it'll be more likely to be passed along, and then eventually everyone will get it because it's such a good mutation. But when we usually think about mutations, we usually think about them in a bad context. But there's probably a lot of mutations that happen, maybe in that non-coding region, that can be neutral, or they can cause no effects. A mutation can make a protein not work well or work poorly. It could make a protein accumulate and then cause problems from that buildup, or a protein might not be made at all. And a mutation can be as little as just one small base pair change in that double helix. Just one small substitution might be enough of a mutation to cause a symptom. But a mutation could also be multiple changes, or it could be missing material or extra material put in. Mutations can be inherited. When we think of genes and genetics, we think of inheritance, but they can also be de novo. They can even vary. Your genome can vary within yourself, depending on when a mutation happens. There's something called a somatic mutation that happens to just part of the body. And that wouldn't be inherited to offspring unless it affected the egg or the sperm. So to show you a visual of this, like say the embryo is formed and it's normal, a somatic mutation happens when the embryo is very small, and then the embryo grows into a person, and then only the tissue that comes from that affected area of the mutation will have the symptom or the bad gene. So in summary, the human body is made of cells. Those cells contain our DNA, and genes are made from the code in the DNA, and those genes are used to make proteins. And the proteins are your building blocks of your body. They make up the tissue and all our organs. Inheritance. So the classic forms of inheritance that, again, in our high school biology classes, we had to learn how to draw these trees and learn about classic forms of inheritance. So there's autosomal dominant, which means you only need one copy of a gene from one side to have that, that type of symptom or appearance. It's called phenotype. So like example, brown eyes or brown hair. You would only need one copy from one parent, and it didn't matter what the other parent, what color eyes or hair they had, you would have brown hair or brown eyes. Autosomal recessive would be you would need one from each side, one from each parent to be that recessive thing. So think about red hair. You need a red hair copy from one parent and a red hair copy from both parents, and then put together it's red hair. There's sex-linked inheritance. So something could be on an X chromosome, and maybe if you only need one copy to work, then a female might not show symptoms, but a male would, because the male only has one X chromosome. But most genetics are not that straightforward. Like I said before, polygenetic syndromes are probably a constellation of different gene copies of various quantities from both sides of the family. And then there's also something called epigenetics, meaning it's not actually the gene, the actual like nucleotides or the DNA coding, but it's that structure of the chromosome, the way it's spiraling around, the way it's kinked and folded. Something's different there. That's causing a change in the way that DNA is being used in the body to cause symptoms. So then, like I said before, some mutations are usually not inherited. Some are known to just be sporadic. So some are known to just happen further on in development, or aren't associated with an inheritance pattern, and some are actually more commonly sporadic than actually inherited. And sometimes the presentation or the syndrome will be different depending on whether the mutation came from the mother versus the father. So there are a lot of different things that can happen to change what symptoms are shown. I'm not trying to be terribly confusing, but you may not know this. Not all DNA is what I just described. So there's also something called mitochondrial DNA. So in the cell, again, the center part, here's the nucleus. The mitochondria are a different part of the cell. They're known as the powerhouse of the cell. They're responsible for the energy production. But there's also a form of DNA that is stored in the mitochondria. Mitochondrial DNA is interesting, so it's carried through the maternal line. So when an embryo is made from a sperm and an egg, the mitochondria of that egg is what gets passed along to the next generation. So it's always from the maternal line. So I like to think this is like science balancing out society. So like in society, people tend to get their father's last name. Well, they get their mother's mitochondrial DNA, so it's balanced. Mitochondrial DNA looks a little different. It's ring-shaped. And I think it's important for this audience to know about mitochondrial DNA because diseases and mutations in mitochondrial DNA can often cause seizures and brain problems, hearing, vision problems, slow growth, weakness, movement disorders, and even like a tendency for strokes, things like that. So it does come up in neurology a bit. Okay. So that's my basics on biology. Now I'm going to move on to genetic tests. So I wanted you to understand a little bit about the basics of genetics so you'd understand what these tests are looking at and what they mean. All right, so who is most likely to have a positive genetic test? So people who've had onset of seizures in the first year of their life, people with those developmental epileptic encephalopathies, and people who have neurodevelopmental comorbidities such as autism and intellectual disability. This population is much more likely to have an underlying genetic cause, but that doesn't mean they're the only population worth testing. So 4 main types or buckets of genetic testing right now. I'll start by describing chromosomal microarrays and karyotypes. Those were the earliest tests that we had and the only tests we had probably when I was starting out my training. The microarray is basically looking for imbalances in the genome. The karyotype looks at that big picture of chromosomes. But more recently, we have much more extensive testing available. Really, it's been phenomenal, the growth over the last 10 years. We now have single gene testing looking for specific genes or panels of genes that were known to cause epilepsy. And then there's something now where we can have whole exome sequencing or even whole genome sequencing. So looking at all of the DNA or all the genetic material. More about that chromosomal microarray. It was really the first targeted epilepsy testing, looking for those imbalances. So what do I mean by that? Meaning like an extra chunk of chromosome or missing pieces of chromosomes or big deletions or duplications. I actually have a patient where a chromosomal microarray identified patterns where the copies of the genes between the two parents looked super similar in some places. And what that suggested was that there was a relationship between the parents biologically. And afterwards, the parents found out that they were actually third cousins from each other. So it can reveal things like that, which may be interesting information and affect how people see things a little differently. Overall, the yield is actually not that high in this testing. Looking at all comers, 3% of people will have a positive test. But if you look in that select population, those high-risk people, early life seizures, developmental encephalopathies, autism, intellectual disability, it might be as high as 15%. To give you an example of what positive testing or what a sample population might get, there was a study of 52 infants with severe unexplained epilepsy, so a high-risk population for having positive genetic testing. And 18% had abnormalities. So what an abnormality is a variant. There's a variation in the DNA that is different from what most people have. But only 6% of these babies was that variant clearly the cause of their epilepsy and their problems they were having. Possibly related 4% because maybe it was a gene known to cause something to do with the brain or seizures or something like that, but not clearly a mutation known to cause seizures. And then 8% of the time they found something unrelated to what they were looking for. And so it didn't explain the problem, but found something that was different. The karyotype is that big picture, looking for missing or extra chunks in the shapes of the chromosomes. So maybe a chromosome's missing, maybe a chromosome's extra, or maybe it's shaped in a ring. So often these are syndromes, meaning a collection of symptoms. And one very common syndrome that most of us know is Down syndrome, also known as trisomy 21. And the tri comes from 3. So you see in 21, meaning it's 3 copies of chromosome 21 instead of 2 copies. Another less common, but another epilepsy syndrome that can be identified on a karyotype would be ring 20 chromosome, and there are some other ring chromosomes too, where if genetic material is missing from either end of a chromosome, it'll link together into a circle or a donut, and that's not normal for our nuclear DNA. So that'll come out on a karyotype. Now, specific genetic testing. This is really where it's gotten exciting. So there are over 926 epilepsy genes now, right? And if you do a panel just looking for those genetic tests, it's faster and it's cheaper than doing some other forms of genetic testing. And a panel can look at hundreds of epilepsy genes. Or it could just look at one gene. So say you know what you're looking for. It could be testing just one specific thing. If you're doing a panel, it's good to have a bigger number, so at least 25 would be recommended. And the yield can really vary. Different studies have given different numbers, anywhere from 4 to 54%, again, depending on who you're looking at. It's highest in people with brain abnormalities that are structural, because a lot of those structural abnormalities are actually caused by genetic variants. And then if one person is pinned down, targeted testing can then be done in other family members. I like gene panels because I'm not a genetic counselor. I don't have extensive training in genetics. And these panels are either it's abnormal, it's not abnormal, or we're not sure. So I can counsel my patients directly on what the risks and benefits of this test are. I don't have to refer to a genetic counselor to have it done. So it's a very useful targeted form of testing for epilepsy. You're not going to get secondary findings that are totally out of the blue and really complicated to manage. Whole exome and genome sequencing is something that's only really become available in the last 10 years and really not done on with much frequency until maybe the past few years. The first large study wasn't even published until 2013. And this would include all the rare and newly discovered genes. And the yield can vary, again, 9 to 50% is what's quoted, but it's very expensive. Insurance may cover this one because if it's positive, it stops all the other genetic testing that you could do. So overall, it could save money because instead of doing those 4 tests total, going 1, 2, 3, 4 from cheapest to most expensive, you start with the most expensive first, and then if you're done, you're done, and then overall, the insurance company will save money. It's often done in trios, and what that means is you do the patient and both of their biological parents. It helps explain the results when you have the parent's DNA, especially if the parents don't have the symptom and the patient does have the symptom, then you know if there's anything questionable or funny. If the parent has it, then you know it's probably not related to their symptoms. Even though we're looking at all of the coding DNA with whole exome sequencing, it can still be non-diagnostic. It's negative in half of patients. And it can miss some things too. It can miss deletions or duplications or triplet repeats that could be picked up by that chromosomal microarray. So it may not end the genetic testing odyssey for everybody. So that's whole exome sequencing, just looking at that 1.5% of the genome that codes for genes. Versus whole genome sequencing, that's looking at every single item of DNA in the entire nuclear DNA complex. This is extremely comprehensive, but it's slow and very expensive. And again, the yield's probably not too different from the whole exome sequencing. And because you're looking at all these non-coding regions, you're more likely to get back, well, this is different, but we don't know what it means. So you're going to get a lot of information that you don't know what to do with. And so it can be very hard to interpret and lead you down a lot more future tests to try to figure out, well, is this important? So both whole exome and whole genome sequencing incorporate all the known epilepsy genes, and they're done in these trios with the parents that help figure out the results. And usually the genetic testing company will store the blood or whatever sample was provided, and they'll store the data for future review, and they may reanalyze it over time when new information is required. So it depends on the company and what their policies are, but they may offer updates in 5 years or up to 5 years after initial testing, something like that. But understand they're not perfect, and if they're negative, it may mean that other testing needs to be done. So who or what gets tested? In medical tests, we usually think of blood, and most people probably provide blood samples, but you can actually get good DNA samples from saliva or cheek swabs. So a lot easier for people to do at home, and then they mail it directly into the company. If someone's having an evaluation for mitochondrial disease, they could do blood or a cheek swab, or actually skin, muscle, or liver biopsy. And then if somebody has a somatic mutation, that may mean the actual tissue affected needs to be sent in. So for some that affect the brain, that means you can't do DNA testing unless you get a biopsy from the affected brain. Like I said before, the whole genome testing, whole exome testing, biological parents are often tested. Insurance won't cover it if you do it retrospectively, so you have to get the parents to do it at the same time as the patient generally in order to get it covered. And it really does help interpret the results. And then later down the line, other family members may want to get tested too based on the findings. People may want to know their personal risk or their carrier status for a disease. So like I said, one panel can follow another if the first one's negative, or the original sample can be reanalyzed and new understandings can emerge because this field is changing constantly. Genetic counseling. What a genetic counselor does is they talk to the patient and their family about the risk of testing beforehand. What are the benefits? What are we looking for to get out of the testing? What it's going to tell you, or what it could tell you, and what it can't tell you, and what to expect in the time frame. After a test is performed, they can explain the findings, what's uncertain, what the unknowns are, and whether additional testing is needed or necessary. So what do we expect from these results? What results can we get? Well, you can get a positive test result. What does that mean? A positive test result means there's been a genetic change identified as the cause for epilepsy. If a person's healthy and doesn't have epilepsy, it could indicate that they're at risk for epilepsy. And it could explain the risks of reproduction and how epilepsy might be inherited in a family. What's a negative test? Well, it means that a genetic cause for epilepsy was not identified using the test that was performed. It doesn't mean that there wasn't a genetic cause. And in some cases, it may mean more testing is needed. What I often get back is sometimes a positive test, sometimes a negative test, but then I really often get these uncertain test results. They're called variants of uncertain significance. It means there's something here that's not common in most people, but the meaning is not known at this time. Variants of uncertain significance, or VUS, the result can be reinterpreted in the future when more information becomes available, and then they can be reclassified as being benign, meaning not associated with any disease, or pathogenic, meaning they do cause a disease or a symptom. They can be very frustrating to get because you don't really know what they mean, and sometimes it's maybe somebody has epilepsy of one type, and I'll get a variant of uncertain significance saying that patient has a single copy for a recessive epilepsy syndrome of a totally different type that the patient doesn't have. And so I don't really know, is that changing the type of epilepsy that they have or their symptoms, or is it really unrelated? And nobody can really answer that. So the benefits of getting a positive test result is there are many of them, actually. It might be able to guide treatment, right? So there's some epilepsy syndromes that respond to vitamins or dietary therapy. There are some genetics. Genetics sometimes gives us guidance on when to use or when not to use specific medications, or when we might be able to stop treatment, or when we you should start treatment. And a positive result may suggest that there are associated risks with the epilepsy. So maybe there's a syndrome that wasn't identified, and then we have to look at other organs and do screening tests on other organs for problems that maybe we didn't know about, or look for tumors somewhere. A positive result can help guide reproductive counseling. How likely are my children to get this gene? They can inform prognosis. Is this something that someone outgrows, or do they live with it, or is it progressive and degenerative? Somebody might be able to join a gene-specific clinical trial, and I think in the future, clinical trials are all going to be stratified by genetic makeup. And a positive test result may lead to social connections. I've had patients who got a weird genetic thing that we didn't really know a lot, but then they found a Facebook group and they just thought it was fun to meet people online with the same genetic variant that they had, and they found that fulfilling. So that, to me, is kind of a surprising good outcome from a positive genetic testing result. Just to give you an idea of when genetics guides management, like I said, there's over 900 epilepsy-related genes, and these are the few where the gene clearly can guide management. So some tell you the specific vitamin B6 or folinic acid that's needed to treat it. There are a few that tell you to use specifically dilantin and phenytoin and other types of drugs like that. And then there are a few that say not to use those same exact drugs. And I just listed here the few that really tell you which medication to use. Mostly to give you an idea that yes, sometimes it guides management, but this is a list of I don't know, 12 genes out of 900 where it clearly guides management. So the likelihood of getting this is actually pretty low, and most of these we suspect very early on in the presentation. So why are results not exact answers? There's genetic profile variability. Right? So this comes in two main forms. One is called phenotypic pleiotropy. This means a single gene mutation, which is known as a genotype, can cause different syndromes, different symptoms from it. These are known as phenotypes. So single genotype can cause multiple phenotypes. For example, there's the SCN1A mutation that's a well-known cause of epilepsy. It can cause just febrile seizures. It can cause febrile seizures that then results in epilepsy. It can cause a condition called migrating seizures of infancy, or Dravet syndrome, which is a really bad epileptic encephalopathy. Or it can cause familial hemiplegic migraine, which is not even epilepsy at all. It's a migraine syndrome. So you could have the same mutation in the same family members, and one has febrile seizures, another has migraines, another has bad seizures, another has benign seizures. And it's the same gene, but it's different in everybody. And we don't have all the answers to explain why someone presents a certain way, or if they have that gene, which symptoms they're going to get. The opposite of that is genetic heterogeneity. So a single clinical syndrome or phenotype has multiple genetic causes. That's probably the case for juvenile myoclonic epilepsy. There are two genes that have been out for a long time, and three more recent ones that were identified that cause juvenile myoclonic epilepsy. But most people with this very distinct type of epilepsy that has myoclonus and sometimes generalized convulsions, sometimes absence seizures, most people don't have positive genetic testing because we're just not there yet, and we think it's probably polygenetic for most people. So what are the risks versus the benefits? Well, the benefits, like I said, they can improve treatment. Some results can guide management, can tell you whether seizures are likely to recur after a long period of seizure freedom, and maybe open up research studies. Maybe the condition identified is life-limiting and palliative care can be thought of earlier rather than later. Maybe extra precautions can be taken for those identified with something like Dravet syndrome, which is known to have a high risk of SUDEP or sudden unexpected death in epilepsy. And knowledge is power. It answers the why. A lot of us just want to know why. It can help with prognosis. It can give access to advocacy groups, peer families, family members, and family planning. It can all be helpful for. A note about the family planning, things to think about is subsequent pregnancies. There can be prenatal testing through amniocentesis or chorionic villus sampling early in the pregnancy to look for the genetic mutation. There's pre-implantation genetic testing through in vitro fertilization. And then people can think about financial preparations. What if my next kid has the same thing? Because we know this is a 50-50 chance with our genetic mutation. And then maybe other family members can get tested to plan their families. So what are the drawbacks? Well, cost, and it maybe need to be repeated over time, so multiple costs. And it can lead to additional testing, which is time, money, and stress. The psychological impact can be really hard to predict. One of my best friends, she got genetic testing, not for epilepsy, but to look at her cancer risk because she was concerned about her family history and she was about to have kids. So about 5 years ago, she got tested and she was told she didn't have any of the known cancer genes. And so she breathed a sigh of relief. She had her 2 kids. And then last year, she was contacted saying that her profile had been updated and they now found a cancer gene in her. But now she's already had her 2 kids and she was going to get her mammograms and her screening tests and colonoscopies and all those things. And so she says, okay, I have a high risk of cancer. What do I do? And they say, well, nothing different. We just know you're at high risk now. So she kind of regrets having had it done, and you can't unknow it once you know it. So there is that risk that should be thought about before you head into this, and that's where genetic counseling can be very helpful. Maybe you don't want to know that there's a poor prognosis, and like I said, there's a lot of variability. So knowing that there's a gene associated with something bad, but maybe we'll be the different ones and not get the bad outcome. It can be hard to handle for some people. There may be a lot of unclear information, which can be frustrating to manage. Risks to insurance, I'll talk about in a little bit. It may affect other family members. Maybe other people in the family don't want to know. And maybe people could be discriminated on based on their genetic testing. Like, I don't know where the future is going. Secondary findings can be stressful and confusing when they're found on testing, especially when you're looking at a lot of different things all at once. These variants of unknown significance can pull people into a long rabbit hole of additional testing and unknowns. Maybe other things were identified that you didn't want to know about. And then again, genotype-phenotype mismatch. Maybe you got a positive test, but doesn't quite match the symptoms. So what do you do with that? It may not be clear. So other people can be affected by the positive results. And so parents, siblings may want to be tested, more distant family may want to be tested. And whether or not it's recommended is based on the diagnosis and the inheritance pattern, and guidance from a medical geneticist or genetic counselor is really recommended. In terms of cost, many children in the US are free. And insurance plans can differ in terms of who's covered beyond that. The plans vary greatly. Usually it needs a pre-approval or prior authorization, but you should find out before the testing's performed. And some will require the genetic counseling be performed before they're going to cover it. The Genetic Information Non-Discrimination Act, GINA, was passed in 2008. It bars health insurers and employers from discriminating based on genetic information, but it only covers health insurance. It does not affect disability, long-term care insurance, auto insurance, or property insurance. Although in 2020, Florida passed, it was the first state to prohibit life, long-term care, and disability insurers from using genetic test results to set premiums, to cancel or limit or deny coverage. Additional challenges. So in VTE, testing was really common. It was the most common panel that I used for a long time because they had a nice epilepsy gene panel. They filed for bankruptcy earlier this year, and now they've been bought out by LabCorp. So a lot of my patients in process there, we have to go through the insurance changes and different authorizations, and where you get your blood drawn is different. So it's caused a little stress in our system. Another challenge is there are huge waiting lists for genetic counselors, for genetic evaluations. There are very few online or virtual options. Counseling may be offered through the testing program. GeneDx is a big provider of genetic testing, and they have counselors that work with them. The only caveat I would say is if you're talking to a counselor about the risk of testing, and that counselor is paid by the company that gets paid for the testing, it seems to me like it might be a little bit biased. And are they really going to go through all the risks of testing that may cause you to back out? I don't know. So the take-home messages, genetic testing is not for everybody. It's a personal choice, and it's guided by medical professionals. It's going to be highest yield in those who've had epilepsy in the first year of life, or those with a developmental epileptic encephalopathy or a neurodevelopmental disability. And the results can affect both the individual and their family members. And the results can have medical, reproductive, psychological, and financial implications. I think it's very exciting. It's a field that's changing constantly, and we're going to have a lot more information and a lot more to do with all this information over the next decade. It's going to be a lot of fun to watch and get a lot of these questions answered. So it is 2024, and I'm not a geneticist. I'm an adult neurologist, but I'm happy to answer any questions that you guys might have about epilepsy-related genetics now, based on what I know of the literature right now. And that's the end of my talk.
Thank you so much, Dr. Bunch. Does anybody have any questions? You can unmute or you can throw them in the chat, whatever works best.
I guess I'll go first. Thanks, Dr. Bunch, for your explanations. It was actually really good and interesting. So based on what you went over, does that mean that gene therapy might be the next frontier for treating epilepsy instead of medications per se, if that makes sense?
Yeah, that's a really, really good point. I didn't bring it up just because it's definitely not there yet, but gene therapy probably will be a part of a lot of things. I think tuberous sclerosis, if you guys are familiar with that condition, probably will be one of the earlier epilepsy syndromes to see sort of genetically guided therapy. It's already had more genetically targeted treatments with medications. I think gene therapy is a real thing and probably will be coming down the line, but it's going to be complicated and it's going to take some time.
Thank you. I just got a direct question as well. If you see siblings with generalized epilepsy and no other neurological diagnoses, would you think that there's a genetic component?
Yeah, so I usually think that there is a genetic component. Whether genetic testing will be helpful at this point, I definitely would offer it to people who have that genetic, kind of like that pattern that suggests there's a genetic cause. But it may not be positive because of like what I said before. A lot of those are probably polygenetic, and there's so much extra information in there that in multiple different genes that the answer may be negative at this point. So perform the genetic testing now, but with the caveat that maybe in 10 years we should repeat it if it's negative, if they want to know.
Beautiful, thank you. The next we have, why and how does someone develop epilepsy later in life?
Yeah, so I think it's interesting because people can be born with even, like I say, a structural abnormality. One could be a focal cortical dysplasia. What that is when the brain is forming, the outer parts of the brain don't quite make it to the destination they're supposed to. So it's a structural abnormality that for some reason often doesn't act up in childhood or even young adulthood, but then can act up when somebody's in their 30s or 40s and becomes symptomatic with seizures. And some of those are genetic. And so somebody in their 40s is saying, you know, I have a genetic problem. Well, yeah, but it didn't act up until now. And people can be born with other structural things like that don't act up till later in life, like blood vessel abnormalities. As people get older, more like in their 50s, then we worry more about tumors, and that's where the imaging becomes more pressing, getting a good quality MRI, making sure there's no brain tumor. And then as people get older, and if they have stroke risk factors, strokes become a big cause. anytime in life, trauma can happen. A head injury can cause seizures. So we see that at any time in life. And then late in life, with neurodegenerative diseases like dementia, like Alzheimer's, we start to see epilepsy sometimes along with that at a higher than normal rate. So developing epilepsy even in older age is actually not uncommon, but the causes are kind of all over the place. Anything really that insults the outer layer of the brain can cause seizures.
As a follow-up to that question, when would a genetic test be recommended for someone who got that diagnosis later in life?
So usually epilepsy later in life, I would look more at the structural abnormalities, the blood vessels, the tumors, the malformations, and look hard for those. I don't often recommend genetic testing for epilepsy that starts after 50. But if somebody's interested in that at that stage, if their development had been normal up until that time, then I don't think the karyotype or the chromosomal microarray would reveal anything, because those would be associated with other symptoms that would have come up by then. So I would recommend going either with the epilepsy gene panel or the whole exome sequencing, and probably which one you would get would probably be determined on what was available to you or possibly which one your insurance would cover.
Beautiful, thank you. The next question, are you familiar with CACNA1A epilepsy and how mild or severe symptoms typically present?
So I'm not highly familiar with that specific mutation and syndrome. I'll just say that I do see a lot of variety in my patients in terms of when they come back with positive testing, and there's a lot of variations on the theme. I think actually that mutation, bringing that up is a great point, because I didn't mention that in that CACNA1A, like a lot of these genes look like random letters and numbers, but what it is actually code for what the problem, like, so CA often stands for a calcium channel problem. And what to me is really fascinating is that our brain works on ions and like these elements going in and out of the cells. So calcium goes in and sodium comes out and potassium goes in, and anything that changes the flow of those electrolytes can cause seizures. So that calcium channel, the mutation in that calcium channel, that it causes seizures because it messes up the flow of calcium in and out of the cell. And then you'll see like letters and numbers after that that says calcium channel, and usually it's talking about a specific mutation, like where in that gene, in which type of substitution or mutation it is. And so while it looks like really random letters and numbers, there's like a code in there. So you can have like SCN1A would be a sodium channel, and then there's like 2A and 8B, and those are all variations of sodium channel gene problems. But yeah, no, I don't have a lot of experience with that specific one. A lot of my patients never got genetic testing when they were a kid, and then now that they're adults, they're not interested, or maybe their family's not available to counsel on whether to get genetic testing. So, but you know, how, like, there are support groups like for people with that specific mutation, which is just, it's amazing. It's great, and you'll get more specific and people who are really familiar with it by going to that specific support group when your local doctor just doesn't get it, because I don't have more than a handful of people with it.
It's great. And I'd also like to add the Rare Epilepsy Network is also a really great resource that can guide you to support groups like nationally, which are, it's essentially made up of a lot of parents who had kids with some sort of genetic epilepsy that they knew more than their doctor after going to many different specialists. So there's a lot of really great resources with the Rare Epilepsy Network as well. All right, any other questions for Dr. Bunch? Alrighty, I'm not seeing any. If anybody has any questions afterwards, you can absolutely shoot me an email or call our office and we can get you connected with Dr. Bunch. And this recording will also be available on YouTube within hopefully by the end of the week as well. So thank you so much, Dr. Bunch, for your time today. This was super fantastic and really helpful information.
Thanks everybody for coming. Happy to share. Have a great day.
Thank you.